The translational analysis, as I stated, was part of a pre-specified set of hypotheses that were designed at the time of the original study design. And we had hypotheses for each of our collections. So at the outset, one overall objective of the study was just to prove feasibility of doing this within a large national cooperative group study. And that I think we did show. I mean, here, what I mean by feasibility is just technical feasibility of being able to collect plasma over a large number of sites within a cooperative group framework, and to be able to actually conduct science on those samples. And I think we did show that we were at least able to achieve that from a technical perspective. In terms of the scientific hypotheses associated with each of the endpoints, there were 3 sets. So the pre-treatment or baseline specimen was analyzed for the amount of tumor tissue modified HPV DNA in the sample as quantified by fragments per ml. And we were looking for a correlation to overall tumor burden. And what we found was that there was a weak to moderate correlation to overall tumor burden, and it did, or what we call gross tumor volume. And it also seemed that there were separately weak to moderate correlations, both with increasing T classification as well as N classification. For the other thing to note about the pre-treatment specimen was that we had a 10% undetectability rate, and I know there's been a lot of interest in what that means. It's probably a combination of factors, with some patients who are P16 positive not truly having HPV etiology, as well as potentially inadequacy of collected archival specimens. But it's also possible that there may be some scientific reasons related to either the HPV type of infection or the form of infection, whether integrated or episomal. That is, there's room for further analysis there to sort some of that out, although I will say it's a small number of patients that were undetectable. And among the patients who were detectable, I think the other thing that a lot of people have been interested in is that we saw about a 6.6% detectability of HPV types other than HPV 16. And I think that was interesting and perhaps a little bit surprising to some people. For the second set of hypotheses, which had to do with the intra-treatment specimen, so this was collected after 2 weeks of protocol treatment, what we were looking at here was whether we were going to have continuous decreases across the course of treatment, or whether the pattern would be more variable. And what we found was that while if you looked at just the median values, yes, it was true that the detectable fragments per ml did trend downwards over the course of treatment, and most patients reached undetectability by the end of treatment collection. But there was variability in the amount at that intra-treatment time point, with some patients showing increases and then subsequent decreases, and some patients, of course, showing patterns of increase. So the number of patients who had detectable fragments per ml at the end of treatment was pretty small, but they did have a noticeably different pattern, of course. And then, like I said, there was this variability to intra-treatment that is sort of interesting to think about in terms of the patterns of response and apoptosis that might be in operation there. The third set of hypotheses had to do with the post-treatment sample, which was collected at a median of 25 days or so after the end of treatment. And I think a lot of people are very interested in that because it's the actual correlation to the eventual outcome. And what we did find was that the negative predictive value overall, just speaking in terms of whether it was total fragment clearance or undetectability as defined more formally, was about 93 or 95%. Correlation with outcomes for the negative predictive value, as well as the clearance rates, and then also that when formally tested for statistical significance, just barely missed the cutoff for local regional control, but for 2-year progression-free survival, there actually was a statistically significant association with continued detectability at the end of treatment. And it seems like a lot of people are very interested in that, as a prognostic significance. Overall, I think one of the really important points from this study is that we've now confirmed that this may be a prognostic biomarker in patients who are treated with radiation-based treatments. It was done in a de-intensification framework, which, as everyone knows, is of great interest in this population. And we did it in a national, large-scale cooperative group trial.